Hello everyone, hope that you are all fine at home. So today we are going to learn about bacterial genetics. So what are the basic principles of bacterial genetics? Bacterial genetics deals with the study of heredity and gene variations seen in bacteria. Bacterial DNA, it possesses a single haploid chromosome comprising of supercoiled circular double-stranded DNA of 1 millimeter length. So DNA is present inside the chromosome as well as extrachromosomally, that is in plasmid. And here DNA lacks basic proteins, and bacteria, they don't possess a true nucleus. Rather, the genetic material is located in an irregularly shaped region called the nucleoid. So the structure of DNA was first described by Watson and Crick. Hence the Watson and Crick model came into play. It is composed of two strands of complementary nucleotides coiled into a double helix. So each strand has got three elements. One is the deoxyribose sugar, which forms the backbone of the structure. Then we have the nitrogenous base and a phosphate group. Now what is the difference between a nucleotide and a nucleoside? A nucleotide consists of sugar, nitrogenous base and phosphate group, whereas a nucleoside lacks phosphate group. There are four bases: two purines, which are adenine and guanine, two pyrimidines, thymine and cytosine. Pairing. Nitrogenous bases of two opposite strands of DNA are held together by strong hydrogen bonds. There are two rules of pairing. First is that adenine of one strand binds with thymine of the opposite strand by forming double hydrogen bonds, and guanine of one strand binds with cytosine of the opposite strand by forming triple hydrogen bonds. DNA replication. In eukaryotes, two strands of the double helix will unwind, they separate out. Each acts as a template for the production of a new DNA strand, and this is done through complementary base pairing. A binds with T and G with C. So there are two mechanisms by which replication can occur: bidirectional replication and rolling circle mechanism. Here I have given the pictorial representation of both the mechanisms. So bidirectional replication, here we have got an origin. This is where the DNA replication starts or begins. The DNA helix is unwound at the replication fork, and both the replication forks, they move outwards from the origin until they have copied the whole replicon, and this forms a theta shaped structure. Coming to rolling circle mechanism, this occurs during bacterial conjugation and during reproduction of virus and bacteriophage. So here the outer strand is nicked and the 3' end of this strand is extended with the help of replication enzymes like helicase, which helps in unwinding of the DNA, topoisomerase, which helps in relieving the tension of the replication fork. Then we have DNA polymerase and DNA ligase. At the same time, 5' end of the outer strand is displaced and it forms a single stranded tail. Now coming to bacterial RNA. Bacterial RNA is similar to DNA, but it has got few differences. Instead of deoxyribose sugar, here we have the ribose sugar. And among nitrogenous bases, uracil replaces thymine. So instead of adenine binding with thymine, here adenine binds with uracil. Polypeptide synthesis. We have learnt in grade 12 as well as in our first year about polypeptide synthesis. So the genetic code, that is a codon, is a sequence of 3 nucleotide bases present on the mRNA, and this contains the information about amino acid synthesis. So in total we have 64 codons, among which 61 are sense codons, that is, they code for amino acids, and 3 non-sense codons or stop codons, UGA, UAG and UAA. Start codon AUG is the first codon of mRNA. Now bacterial variation. It is of two types, phenotypic and genotypic. Under phenotypic, the variations in the expression of various characteristics of the bacterium is seen. Genotypic, there is a change in the genetic constitution of the organism. Mutation, it is a random, undirected, heritable variation caused by change in the nucleotide sequence of the genome of the bacterial cell. It occurs in two manners, spontaneous and induced. Spontaneous, it occurs naturally in dividing cells, whereas induced, it is caused as a result of exposure to a mutagen. Mutagens can be physical or chemical. An example for physical mutagen is UV radiation and chemical is acridine dyes. Mutation is of two types, small scale and large scale. Under small scale mutations, in bacteria we see point mutations or addition or deletion of a single nucleotide pair, whereas under large scale there is deletion or addition of multiple nucleotide pairs. How can mutants be detected? We can detect mutants by a number of tests: fluctuation test, replica plating method and Ames test. Now coming to gene transfer. There are two types of gene transfer, vertical and horizontal. Here we are going to concentrate on horizontal gene transfer, that is transformation, transduction, lysogenic conversion and conjugation. So transformation. In this, the bacterium takes DNA which is free, that is, it is naked or free DNA present in the surrounding medium is taken up by the bacterial cell, and this is incorporated into its chromosome in a heritable form, that is, its progeny can inherit this variation. So the process goes as follows. Long dsDNA comes in contact with a competent bacterium, binds with the DNA binding protein, and one strand is degraded, the other strand will associate with the competent specific protein and gets internalized. This is how it gets integrated into the host chromosome. So Griffith's experiment, it provides the direct evidence of transformation. Transduction, it is a transmission of a portion of DNA from one bacterium to another by means of a bacteriophage. So there are two types of transduction, generalized and restricted or specialized. So the main roles of transduction are in drug resistance and for treatment purpose as a method of genetic engineering. Lysogenic conversion. In the lysogenic cycle, the host bacterium is unharmed. So here the phage DNA remains integrated with bacterial chromosome as prophage. So they both, the bacterial DNA and the prophage DNA, both of them will multiply synchronously. This imparts toxigenicity to the bacteria. Here the phage DNA itself behaves as the new genetic element. So this is just a representation of Griffith's experiment about transformation. Here the S strain, smooth strain, is injected into mice, it dies. When the rough strain is injected, it lives. Whereas when a heat-killed smooth strain is injected, it is alive. When heat-killed smooth strain and rough strain are injected together, the mice dies. Okay, coming to the two types of transduction, which are generalized and specialized. Under generalized, the phage DNA enters the bacterial cell. The host DNA is hydrolyzed into pieces and phage DNA proteins are made. Occasionally, bacterial DNA fragment is packaged in a phage capsid. The transducing phages infect new host cells, recombination takes place, and it further grows, the bacterial cell grows. Now coming to specialized or restricted type of transduction. Here the prophage DNA, the initial steps are similar, but later on the prophage DNA will exit incorrectly or by mistake. So it takes the adjoining bacterial cell along with it. This is how transduction or the host DNA is transferred. Now coming to conjugation. Transfer of genetic material from one bacterium to another by mating or contact, that is by formation of a bridge or a conjugation tube between the two bacterium, is known as conjugation. So there are different types. First is F positive, F negative mating. So the F positive cell, it has got a plasmid called F factor or fertility factor. The one which lacks this factor is known as recipient or F negative cell. F factor conjugation plasmid, it carries genes which encode information of the sex pilus, whereas F pilus, it brings both the cells, that is the F positive and the F negative cells together and forms a bridge between them. Here replication takes place by rolling circle mechanism. The copy moves into the F negative cell through the bridge, and when this copy, that is when the F factor moves into the F negative cell, it is completely transformed into a F positive cell, and therefore it can further conjugate other F negative cells. HFR conjugation. HFR cells are those which have got high frequency of recombination. The F factor being a plasmid, it may integrate with the bacterial chromosome and behave as episome. So during conjugation between these two cells, that is HFR and F negative cells, only few genes are transferred because the bridge is broken before the whole genome is transferred. Hence, an F recipient cells are never completely transformed into F positive cells. F prime conjugation. F positive cell to HFR cell conversion is reversible. F prime factor is a factor which has got some chromosomal DNA. Sexduction. When an F prime cell conjugates with F negative cell, it transfers the host DNA incorporated with it along with the F factor. The recipient becomes F positive. Now coming to carcinogenic factor or call factors. So bacteriocin production in bacteria is plasmid coded, which may be transferred by conjugation. So those plasmids which transfer this bacteriocin through conjugation are known as call factors. Conjugation is also an important method of transferring plasmids coding for multiple drug resistance among bacteria. Fate of donor DNA following horizontal transfer. So once the donor DNA enters the recipient cell via the horizontal transfer methods like transformation, transduction, etc., a merozygote is formed. So what is the fate of this cell? Either it gets recombinated with the recipient chromosome, or it becomes a partially diploid cell, or there is host restriction, that is, it is not recombined with the recipient chromosome. It is restricted by the host cell chromosome. Bacterial recombination. It is of two types, general or homologous and site specific. General type, this is the most common, and this occurs at any place in the recipient's chromosome between homologous sequences. So the rec genes belong to the recipient's chromosome. Crossing over occurs, that is, donor and recipient DNA strands will break and they further rejoin. Whereas in site specific, donor DNA is not homologous with the chromosome it joins, and the enzymes involved are specific for the particular bacteriophage and host bacteria. Under general type of bacterial recombination, there is a holiday model which is divided into two, reciprocal and non-reciprocal. Reciprocal exchange between a pair of homologous DNA sequence between the donor and recipient strands occurs in case of reciprocal type, and in non-reciprocal, the single donor DNA strand is inserted into the host chromosome by replacing a piece of host chromosome. That is, a piece of host chromosome is cut off, and where it is cut off, that is the place where the donor DNA rejoins. Now coming to transposition. Transposons are bacterial genes which are capable of intracellular transfer between chromosome to chromosome, plasmid to chromosome, plasmid to plasmid, and this process, it takes place with the help of transposons. It is also called jumping gene or mobile genetic element. It does not require any DNA homology. So transposons are classified into two: insertion sequence transposon and composite transposon. Insertion sequence transposon, it is the simplest form, the length ranging from 1 to 2 kilobase pairs. It has got a transposase gene and it is flanked at both the ends by an inverted repeat sequence of nucleotide. Composite transposon, they are larger ones and they have got additional genes. They code for antibiotic resistance and toxin production. Both ends are flanked by the insertion sequence. Now coming to the last topic, that is gene transfer by artificial methods. It is done by three ways. That is, first, genetic engineering. It is a deliberate modification of an organism's genetic information by directly altering its nucleic acid genome, or DNA technology. So first, the DNA is treated by restriction enzymes. It gets fragmented and then southern blotting is done. So in a capsule, southern blotting, first it begins with electrophoresis, then it is transferred to a nitrocellulose membrane. The desired gene is obtained and isolated, recombinated with a vector, introduced into bacteria, and cloning is done. What are the applications? It helps in production of vaccine, antigens used in diagnostic kits like ELISA, and for the production of proteins used in therapy, in transgenic animals, and gene therapy. So what is a vector? It is a small piece of DNA into which a foreign DNA fragment can be inserted, and that can be stably maintained in an organism and be used for cloning. Coming to the second method, nucleic acid probe. So radio-labeled or fluorescent labeled pieces of single-stranded DNA or RNA. This is what is known as a nucleic acid probe. So what is hybridization? It is a technique in which two strands, single strands of DNA or nucleic acid comes together and forms a double-stranded molecule. So coming to blotting techniques. Method of transferring DNA or RNA or proteins from gel onto a carrier, followed by their detection by using specific nucleic acid probes or by enzyme immunoassay like in ELISA. So there are four types of blotting techniques. First is southern blot, which is done to detect DNA. Then we have got northern blotting, which is done to detect RNA. Western blot detects proteins, and eastern blot, it is actually a modification of the western blot in order to analyze the proteins for post-translational modifications using probes.